Okay, so this is to a second gram stain animation that's on the internet. So I'm just going to open up that page so that I have it open when I get to that part in my lecture. I'm going to shrink this down and go to today's lecture, which is Lab 5. And so we're going to start out talking about hypotheses, what is sometimes referred to as if-then statements. And then another colleague of mine, like I said, he likes to write stuff mathematically. So it's presented there. You don't have to know it for this course. If that makes sense to you, great. If it doesn't, don't freak out, right? I still have trouble like kind of thinking in writing things in symbols. I'm not really a symbol person, right? Don't do so great with symbols. I need words and analogies, and stories. So we're going to do gram stain today. So as I said, I've already made a heat-fixed emulsion for you guys. You're just going to stain it. It has a circle with both gram-positive and gram-negative. It has E. coli in one of the circles, and E. coli, as we know already, is a rod-shaped bacteria. So on the last page of your guys' handout for today, right, one of the questions is what shape is each of the organisms that's on our gram stain smear, which is going to help us identify them. So E. coli is rod-shaped. It has a singular arrangement. They're not going to stay next to each other after they divide. And then Micrococcus luteus, we already said, is what? Spherical, cocci shaped, and it forms what characteristic arrangement? Tetrads, that group of 4. Oh, staph epi. But actually, I did Micrococcus luteus. So you might want to scratch out staph epi and write Micrococcus luteus. Sorry. Right? So, shape and arrangement. E. coli is a rod, a bacillus, and it doesn't have a characteristic arrangement. It'll be single cells all by themselves. They're not going to form chains or pairs or anything like that. And then for the capsule stain, you'll see there's 4 different specimens that you're going to stain using the capsule stain procedure. And one of them is not going to have a capsule. So your task today, as a class, as a group, as pairs, however you want to figure it out, there's one slide today that you're going to make a capsule stain, and the organism is not going to have a capsule. Okay, so that's the mystery for today. Which one is it? So, how to make a hypothesis and test one. So, a hypothesis can also be referred to as an if-then statement, right? So, if this, then this, right? So, if my computer, as I just experienced today, right, is falling asleep, I know, right, or I hypothesize that it's a power issue, right? So then I can test that hypothesis by figuring out, is there power to my computer? So the first thing I looked at, is it plugged in at the computer? It was. Is it plugged in the outlet? It was not, right? I plugged it in, then I had power, then it no longer went to sleep, right? But we can apply this to what we've been doing in lab. So as we were talking about, as we were reviewing our quiz from last week, A, if, then B, we could say if one morphology, right, one shape, one look to the organisms on that plate, that equals one species. Is that true? No, it's not, right? This is a hypothesis, right, stating that one morphology equals one species. But we know this not to be true, right? We would disprove this hypothesis. We could also flip it another way, right? So if one morphology equals one species, 2 morphologies should equal 2 species. Is that true? Is that true? 2 species equals 2 different morphologies? Sometimes, but Micrococcus luteus and staph, right? Other than the fact that I realized I put Micrococcus luteus on your guys' slides, I forgot your handout said staph. You wouldn't know the difference per se because they're both cocci. Microscopically, they're both the same morphology, but they're two different species. They're two different species. So none of these hypotheses hold true, right? In order for them to be true, both statements would both have to be true or both statements would have to be false, right? So, in this, So in this case, both of them are false, right? So we would not agree with these hypotheses. To agree with them, they would both have to be true, and they're not. Okay? Still with me? Maybe. Pretending. Okay? So the hypothesis is false, too, when one is true and the other is false. Right? So this one could have been true. One morphology, one species. Right? But if two morphologies equals two species equals two morphologies, then we can't accept it, right? Because one is true and the other one is false. They got to match up. Make sense? Okay? So, in other words, a hypothesis is disproved if the first part of the statement is true while the second part is false, right? So, here, for just this one hypothesis, one morphology equals one species, right? It's false, right? And then for this one too, two species doesn't always equal two morphologies, right? So observing one morphology of a mixed culture would disprove the hypotheses since part A, right, is written, is true. Is rewritten as true. All right, I'm not going to read what's on the page because I'm messing it up. If a hypothesis cannot be refuted, right, then we have supporting evidence. Have there been hypotheses, things that we believe to be true, that are later disproved? Yes. Absolutely. And all the time in science, right, a lot of times what we're doing is someone has a particular hypothesis, they say, A equals B, and we go, no, I don't think so. And I'm going to do my damnedest to prove to you that A does not equal B. Right? That if A is this, B is not the reason. Right? Is not the result. Okay? Theories are hypotheses that have very strong supportive evidence to them. Right? When we say something is a theory. Okay? It's usually very widely supported. Does that mean that someday someone's not going to come along and disprove it? No. Right? When I was in school, and you always have to worry about, especially with true and false statements, these all or nothing things, right? In science, it's never usually all, right? There's always an exception to a situation. So in the case of prokaryotic cells, when I was in school, we didn't learn about a cytoskeleton for them, about proteins inside helping them hold their structure, right? I was taught they don't have a cytoskeleton. That's not true. They do. We didn't have the high-powered microscopes and the techniques that we have today back then. So because we didn't see it, we didn't think it was there. But if you think about it, kind of makes sense, right? Why wouldn't a cell have structural support inside? Right? Why wouldn't it? It does. Again, we just could not see it yet. Okay. Yeah. Yeah. And so now you understand, right? So some of you guys are having to come back to school because your prerequisites are out of date, right? Science is continually evolving and changing, right? We learn new information, but guess what always holds true? Scientific method. How we go about, and we do this every day in our everyday lives, right? I did it 2 minutes ago, right? Why is my computer going to sleep, right? I formed a hypothesis. Well, it probably doesn't have electricity. Let me test it. Let me figure it out, right? So for all of the stuff that we do in lab, right, we can go through this process of forming hypotheses. So later on in the semester, we're going to say fluorescence, right, of the bacteria can only happen if we have expression of the green fluorescent protein gene, right? You can't get it without this, right? These two things are connected. So if it doesn't fluoresce, it didn't express the genetic information, it didn't make the protein. If it does fluoresce, right, then it's supported, right, that you can only get the fluorescence with the expression of that genetic information, that production of that protein. Oh, thank you. You can just give it to me.
Sorry.
Yeah, you're fine. You too. So, these two things are linked, right? If, then, right? And the same holds true for the opposite. You can't have one without the other. Make sense? Okay. So, as I said, we do this all the time. Lots of different careers use this other than just scientists, right? Lawyers use this in their arguments, right? You can't have committed a crime without a weapon, right? Or you couldn't have stabbed somebody without a knife, right? So we got to prove that that knife went into that person, right? Engineers, mechanics, and then ourselves, right? In our everyday life, we use it more and more than we realize. We just structure it, right, when it comes to science. We define and follow the steps. So, as I said, unlike some of the others, we're really actually a lot of times trying to disprove hypotheses more so than prove them, believe it or not. So, journal articles, right? Scientific journal articles are set up this way. You have an introduction, right? They're going to introduce their hypotheses. A, if, then B, right? If A, then B. They may even have more than one hypothesis. We ask ourselves, why are we doing these experiments, right? What's our goal? What's the question we're asking, right? What are the outcomes? And then we have our methods, what we're going to actually do to prove or disprove that hypothesis, right? So what are we actually doing? So you guys are using proven methods, right? Protocols already written out for you that you rewrote, right? The series of steps that are going to help us with our experiment. Results, right? You're going to do something, you're going to get a result, right? You're going to look under the microscope, you're going to see pink or purple, or you're going to see a capsule, or you're not going to see a capsule. What are you going to do? You're going to go, oh, cool, this one doesn't have a capsule. Is that it? What are you going to do? You're going to write it down, you're going to draw it, you may even take pictures with your little phones, right? You're going to record your results, right? You're going to make observations and you're going to record them in words, but you may even have figures or graphs or tables or diagrams, right? And of course, you're going to exclude from your analysis if you know you made a mistake, right? So if you realize you skip the iodine step, are you going to trust your results? No. And that's why later, too, like, I know what you're supposed to get for results. These are basically controls I've given you, right? Later on, when you have an unknown organism, to make sure you did the procedure correctly, you're going to have standards, you're going to have controls. You're going to put 3 circles on your slide. You're going to put a known gram positive, a known gram negative, and your unknown. So if your positive and negative controls come out correctly, then you know you can trust your results for your unknown organism, right? Because this is a tough technique, right? Unlike the capsule stain, when you guys do it, right, we're not going to do a positive and negative control for that. You already know what you're supposed to see or not see, and it's not a very complicated technique with multiple steps where things could go wrong, okay? But if you definitely know you made a mistake, you're obviously not going to count that. But of course, if you discovered something new, right, you are going to record that. That happens in science quite a bit, right? You're trying to do one thing and you discover a whole other thing. So, what outcomes should I record and how should I report them, right? And that's important too. Do we need a picture of this? Do we need to draw it? Do we need to describe it? Right? How should we best record these things? And then in the discussion of an article, right, we're going to justify any ambiguous or contradictory results. We're going to draw conclusions, right? So the fact that it came out purple in the gram stain, what does that mean, right? And this is a pretty widely accepted protocol, so it gives us some type of information that's based on previous experiments, right, that this organism probably looks like this if you were looking at it all the way down to the molecular structure. Right? So, what does this data mean? Right? Big whoop-de-doo, it's purple. Right? Why is that important? Right? Why is it important that it's purple? So, Graham, it's actually a guy's name, right? In 1884, came up with this method that you guys are going to use today. So, as you can imagine, it's been done for a long time. So Hans Christian Graham developed this method for distinguishing the two major classes of bacteria. Handsome, huh? It's kind of scary looking if you ask me. So serious. Nobody smiled for pictures back then. Okay, so a 4-step process. In between each step, you rinse with water, right? So the first step is the primary stain, which in this case is crystal violet. We're going to leave it on there for 1 minute. No matter what the animations say, and I can't remember what your book says, 1 minute is the standard rule, remember, in microbiology as far as staining goes. Right? It's pretty easy. You just watch the little red hand go around one time. Right? It's pretty easy to keep track of. Then you're going to rinse the slide. Right? You're flooding the side with crystal violet. You're going to rinse it off with water. Then you're going to do the next step, which is applying iodine. And iodine, in this case, is a mordant. It adheres to the crystal violet. This, too, we're going to do for 1 minute. Rinse with water. And then we're going to decolorize using 95% ethanol. This is the differential step of this procedure and makes this staining procedure actually a differential stain. We're going to apply this dropwise until the purple color goes from a dark purple to a light purple. And this is the tricky step. And this is why, as I said, I'm going to demo this step for you guys. And why I had the audiovisual department make that really good video for us. The last step is the counter stain so that we can stain the cells that have become decolorized in the process. We stain them with safranin. It's a contrasting color from the purple. This is a red dye that will appear pink for the cells that get decolorized and then stained with the safranin. I'm going to do this for 1 minute, rinse, and then we're going to blot dry and then look at under the microscope. So today you guys will see in 2 separate circles, right, both purple and pink organisms, because you have both a gram-positive and gram-negative on your slides. Our gram-negative, just as is seen here, is a rod-shaped bacteria that will be pinkish. And our gram-positive is a cocci, and as I said, it's Micrococcus luteus, so we might even see those tetrads. And so they'll appear purple. Gram-positive and gram-negative in the gram stain don't refer to charge at all. It has to do with, this is like doing a test where we want to know the result. Did it stay purple? Yes, it's positive. It's gram-positive. It stayed purple throughout the entire process. If it got decolorized and stained by our counter stain, it's negative, you could say, for the gram stain reaction. It did not stay purple, right? So therefore it comes out pink. Although charges come into play, but when we're saying gram-positive and gram-negative, we're talking about a result in this test, right? Did it stay purple? Yes, it's pink. It's purple. It's positive. Or did it not stay purple? It's pink. It's gram-negative. So the shape does not determine whether it's gram-positive or gram-negative either. Although in this lab, we work with cocci and rods, none of our cocci are gram-negative. Most gram-negative cocci are pretty nasty pathogens, like Neisseria gonorrhea and Neisseria meningitis. I don't think any of us want to work with that, right? So we don't have any gram-negative cocci. So if you see pink cocci under the microscope, you did something wrong, right? Or you've got contamination that we don't want to have. We have lots of gram-negative bacilli that we'll work with. So for instance, today, Escherichia coli, but also the Klebsiella that we'll be capsule staining is a gram-negative rod. We've got lots of gram-negative rods that we'll work with in this course. And then we have several gram-positive cocci that we work with, like the Micrococcus luteus and the Staphylococcus epididymis. And then there's a few gram-positive rods that we work with. One you've already worked with, which is the Bacillus cereus, Bacillus megaterium, and actually Bacillus subtilis. All the bacilli that we work with that are spore formers too. And you may have seen, remember the spores that didn't stain last time? Next week we'll actually stain the spores from the Bacillus species that we have. They're all gram-positive when you gram stain them. They'll all be purple rods. So now we're going to go, and I'm not going to do animation 1, I'm going to do 2. So it's all about the cell wall composition of these bacteria, is why we get the different colors. So we're going to look at that. So thank you to North Carolina University for putting this together, because this one rocks. There's no audio to this, but of course, if you listen to my recording, then you have my audio. But if you were to look at the link on its own, don't try and fix your audio on your computer. There is no audio. It is annotated in that it does have a script. So down here, right, is their script for each one of the slides. So you'll notice that in the beginning I clicked on gram-positive. So we're going to go through the steps for organisms that would stay purple, that what we call gram-positive. Right? So represented here is the cell membrane, right, with its phospholipids and its embedded proteins. Here we have the peptidoglycan layer, which is unique to bacteria. This is a sugar and amino acid matrix, and then also associated with it for gram-positive cells, this represents the tectonic teichoic acid, which is almost like rebar. Right? It helps support this cell wall layer. Now there's holes between the molecules. Stuff can travel in and out as it needs to for that cell to survive. The membrane is what determines actually what gets in and what goes out. Right? That's the semi-permeable layer. The cell wall layer, anything goes in and out of it. Okay? It's mostly structural support, and it's why we see the different shapes, why we see a cocci or a rod. But the main function of that cell wall is to keep the cell from bursting under osmotic pressure. When water comes rushing into the cell, it keeps it from bursting. So when we do the gram stain, the first step we do is apply the crystal violet. Now remember, crystal violet's a basic dye, so what charge does it have? It's positively charged. So it's attracted to the negative components inside of our cells. And what makes our cells very negative on the inside? Or bacterial cells? No, not negative thoughts. What molecule is highly negatively charged? The membrane is negatively charged. That's the phospholipids. But what inside the cell? For us, it's inside the nucleus. DNA. DNA is highly negatively charged, right? So that positive basic dye is attracted to the inside of the cell, attracted to that DNA. So it's going to go inside. So there's our crystal violet, right through that cell wall. The cells are dead too now, right? We've heat fixed them. We've killed them onto this slide. They can't stop this crystal violet from going in. And in fact, living gram-positive cells can't stop crystal violet from going in either, and it would kill them. Anyways, the stain. So it's going to go in. Then the next step is applying grams iodine. This is going to go into the cell as well. And this is actually going to bind with the crystal violet and create a much larger molecule, what we refer to as the diiodine complex. So now it's bonded together. The next step is decolorization with ethanol. And what happens here the cell wall shrinks. The bonds between the molecules shorten, right? And the holes between the molecules get smaller. So it shrinks up. Because that happens, and these holes just became smaller, it traps in the crystal violet iodine. This doesn't get out of the cell easily. So these guys are going to stay purple. But for the gram-negatives, they would be clear or colorless, as you'll see in a moment. So in order to see them, the last step in the procedure is adding safranin. So the gram-positives are going to get stained by this safranin as well. So that's going to go into the cells as well. So my best analogy of this is laundry, right? If you stick a red sock in, which I did once when I was a kid, with the whites, your dad ends up with pink underwear. Right? It's great. That red runs onto the white, right? But if, say, you had black underwear, right, and you threw in a red sock, would you notice if it bled onto it? No, because that black underwear, you can't see that it's picked up the red dye because it's so dark to begin with. So in this case, they're purple, right? So again, imagine purple underwear, right? If you throw a red sock in, are you going to notice that the red sock bled onto the purple underwear? Probably not. For the most part, you're just going to see purple. So the same is true for these cells. They're just going to be a darker shade of purple, right? But they're still going to be purple. Make sense? So for our gram-negatives, oops, let's see. It'll go back to the main screen this way. So under the microscope, they're going to look purple, right? And they could be cocci, they could be rods, right? They could be other shapes. The shape has nothing to do with what color they are. It's about the cell wall, whether they have that thick peptidoglycan layer or not. So for gram-negatives, their structure is different, right? Notice we've got more layers here. We've got the cell membrane. We've got a little bit of peptidoglycan here, not quite as much as the gram-positives. And we've got another membrane called the outer membrane. And this is a very special layer too, in that this very top layer, these are not phospholipids. This is lipopolysaccharide. So there's still the phosphate in the fat, but here are sugars associated with it. And the bad news for us is that gram-negatives, when they break up and die, this is toxic to us. Their outer leaflet, their LPS, their lipopolysaccharide, is toxic. It's what they call endotoxin, because it's basically from the cell, within the cell. Right? When the cell's alive, they don't release these, but when the cells die, they release them. So even when we give people with gram-negative infections antibiotics, sometimes we can make the condition worse because we're killing cells and releasing these toxins as we're killing the cells. So you have to be very careful with gram-negative infections, especially when they get into the blood. And we can detect for these components when people have infections. And it's bad news, right, when you see these at the blood level. So these guys, because of the different layers and what they're made of, they're going to respond differently to the steps in the process. So the first step is always the same. It's still crystal violet. It's still going to get into these cells because, again, they're dead, where when they're alive, that outer membrane would actually stop the crystal violet from getting in. Then the next step is the iodine. It's going to go in. Same thing's going to happen. It's going to bind with the crystal violet, make that larger molecule. Then, when we decolorize, watch what happens. Oop. We lost something, didn't we? The outer membrane is gone. Fats, lipids are very soluble in ethanol. Ethanol dissolves them. And this is why the hand sanitizers and stuff like that you use that contain ethanol, alcohol, or isopropyl alcohol are effective because they damage the outer membrane of gram-negative bacteria and really affect them. It did shrink the peptidoglycan layer, but the layer was very thin. And so, therefore, it doesn't really trap the stuff in. It gets washed out at this step. So this is why we're going to have purple coming off of our slides. So they're colorless at this point under the microscope. So remember, bacteria are relatively translucent unless we stain them. But we just removed the stain. So now we've got to stain them again so we can see them. So we counter stain with a contrasting dye, in this case safranin. Because these guys have undergone some damage, they're not going to be red, right? They're not going to hold on to a lot of the red dye. They're instead going to appear pink under the microscope. And therefore they're referred to as gram-negative, because they didn't stay purple. They're not gram-positive. They're gram-negative. Make sense? All right. So, and then you could start all over again. So it is that cell wall, especially for gram-positive, that very thick layer, peptidoglycan, that traps in the crystal violet iodine. That's going to allow these cells to stay purple when we stain them. So gram-positives, right, really thick layer of peptidoglycan. Gram-negatives, thin layer. And that outer membrane, which is what gets damaged by the ethanol treatment, the decolorization step, and actually decolorizes those cells. So I don't know if they did this on purpose or what, but kind of bluish, kind of reddish. So as I said earlier too, the purpose of the cell wall is actually keep the cells from bursting, right, under osmotic pressure. And that tectonic acid, as I said, creates additional structure because for gram-positive cells, they have multiple layers of peptidoglycan. So because there's so many layers, it would be kind of wobbly if you didn't have additional support. Just like when we stick rebar in when they're pouring cement, right? It helps stabilize the thickness of that cement. So gram-negatives are usually only a few nanometers thick of peptidoglycan, where it could be anywhere from 30 to 100. But remember, the resolution of our microscopes is only 200 nanometers. So if anything, right, that whole outer covering, the membrane, the cell wall, would just look like a line under the microscope, right? A very thin line at that. So we only know this type of information from much more high-powered microscopes, like electron microscopes, where we can see the 3 distinct layers for gram-negatives and the 2 layers for gram-positives. So peptidoglycan is sugars, right? 2 modified sugars. Sometimes the substance is referred to as murine. It's N-acetyl glutamine and N-acetyl muric acid are the 2 sugars, and they alternate in linkage to each other, and then they're linked to each other by amino acids. So that's the peptide. Peptide is the name of the bond between amino acids. Glycan refers to the sugars that make up the peptidoglycan. Glycoproteins, therefore, right, are sugar and protein. So you have your protein component with the sugar attached to it. These are very common in biological systems to have sugars attached to proteins. They love each other. So why is the crystal violet iodine complex bigger than just the crystal violet chlorine? Because remember that it's usually associated with chlorine. When we put it into solution, they dissociate, right? But both of those are present in the dye that we call crystal violet. A little bit of chemistry, not too bad, right? On the periodic table, they're in the same column, right? Here is chlorine right here. Here is iodine. When you switch one out for the other, right, and they bind to each other inside the cell, it's lower on the periodic table. It's larger molecule than chlorine. Iodine's bigger, right? So chlorine has 17 protons and 17 electrons, whereas iodine has 53 protons. And that's a typo, I think, because it should also have 53 electrons. Well, then it usually gains an electron, so in that case it would be 54. That's right, and 58, because it becomes negatively charged. When it's not charged, right, it has 53. When it's charged, it grabbed an electron from crystal violet, right? So this is Peter's cat, his stray cat that he's adopted. So she can fit through the little gap in the fence, right? When she comes to eat, after she eats, she can't leave. She can't fit through the gap anymore in the fence. She has to stay around for a while, but she pretty much stays now, right? I think she's adopted him more so, right? But just that small change of eating, right? She can't fit back through that gap. What were you gonna say, Tyler?
Was that vaccinated by chance?
Oh, I don't know. You'd have to ask Peter. It's his stray cat. He lets her in the house and everything. He's probably taken her to the vet. But so these are a couple toys, right, that help us understand this phenomenon of how two things together are bigger than just one alone. So this is a common toy that most kids play with. I don't have one still. I wish I kind of did. So then I bring it in for you guys. But this has two halves, which you can pull apart, and the little blocks that are inside fall out. And the little kids put the blocks in, and these blocks barely fit into these little shaped holes. Little kids usually don't know unless you show them, right, that once you put the blocks in, you got to pull the two parts apart to dump the blocks back out. Well, they don't know. They put all the blocks in, they want to do it again. So they stick their little hand in one of those holes and grab onto a block. Well, the problem is that block barely fit in that hole on its own. Never mind with a hand around it. So then the kids start screaming, and you have to convince them to let go of the block so you can get their little hand back out. And then you can show them how to open it. But, it really drives home that part that, that block and the hand are not coming back out that hole. Well, in the case of the gram stain, y'all, this is a terrifying situation. Because in the case of the gram stain, the crystal violet goes in, right, like the block goes in. Then the iodine goes in like the hand goes in. And then what happens to that hole when you decolorize? It shrinks. Could you imagine sticking your hand in and then the hole shrinking around your hand? You'd really have a screaming kid. Right? But that's what happens in the gram stain, and that's why, right, the crystal violet iodine doesn't come back out. Why you can't get a hand and a block back out that hole. So a little intelligence quiz for you guys. How do you get that ball out the hole? Those you guys in lecture on, you know the answer. Right. So that ball is not going to fit out that hole, right? Not the way it is. What do we got to do to it? We got to remove those pegs, right? Those pegs are removable, right? That's an intelligence quiz, believe it or not. This little puzzle right here. Okay. All right. So we could go step by step, right? Tables are a good way to organize information, right? I highly suggest that you use them. So step by step, right? What happens, right? This is just all written out for you guys. I'm not going to read it again, right? I've already pretty much said it, right? And then for gram negatives, and then together. And this is the important step, right? This is what makes things different, is the decolorization step. Right, removing that color from our gram negatives, the ones with that outer membrane, and then colorizing them again with the safranin. So this is just diagrammically another way of representing the information, right? So we have two cells, we stain them, the crystal violet, we add the iodine, we decolorize the gram negatives, and then we counterstain, and we even made this one a little bit more purple, right? So in fact, they become a darker purple. And one of our questions for our homework, which is to predict the mistakes and how they would affect, one of them asks if you reverse the order, how would it look? And I've actually done this to see what it would look like. And it'll make sense why that darker purple comes into play. So we'll think of each one of these separately. So let's review. What are the four major steps? Step 1, crystal violet. Step 2, iodine. Step 3, ethanol decolorization for safranin, right? So for these mistakes, we're just talking about one mistake, right? You made this one mistake. So if you forgot to add the iodine, but you did all the other steps, what would gram-positives look like and why? So let's look at each step. So you still did the crystal violet, right? So they're going to be purple. You didn't do the iodine, right? So what did we not form? The crystal iodine complex, right? So what's probably going to happen when we decolorize? They're going to become colorless because we don't have that big molecule now. We just have little crystal violet by itself. It's going to escape out those holes because it's small. So then we're going to do the next step, which is staying with safranin. So what color are our cells going to be? Pink. Are gram-positives supposed to be pink? No. Will gram-negatives come out as expected? So if we forget the iodine step, are gram-negatives going to come out pink? Yeah, they still will. Because remember, this step isn't really important for them because we wash out the crystal violet iodine complex, right? So no big deal. So what? They get decolorized. They're supposed to get decolorized, right? We are supposed to stain them with safranin. They are supposed to end up pink. So this is a correct result. The problem is that both of our cells right now are what? Pink. They're both pink. Gram-positives and gram-negatives are pink because you forgot the iodine. What happens if you forget to decolorize? Mm-hmm. Okay, you want me to go back? I can go back. Yeah, so for gram-negatives, we're going to make them purple. No iodine, so no crystal iodine complex, which doesn't really matter for these guys because they get decolorized anyways. Right? So it gets decolorized, and then we counterstain, it becomes pink. Like it would normally do anyways. Now what happens if you don't decolorize a gram-positive cell? Is that going to make a difference? No, they don't get decolorized anyways, right? They're going to be purple, they're going to be crystal violet, they're not going to get decolorized anyways. They're going to get stained with safranin, except now they're going to be a darker purple as opposed to a lighter purple. Can you tell the difference between dark and light purple?
A little bit.
If you have something to compare it to, you can. Right? In this case, you wouldn't. But does this create a problem for our gram-negatives? Absolutely, right? They won't end up being pink. They'll end up being what? Purple. Purple? Well, let's go through the steps. You're going to make them purple, right? You're going to add the iodine. They're still going to be purple. You're going to decolorize them. They're going to be, oops, we forgot that step. So they're going to be purple. Then you're going to stain them with safranin, so they're still only going to be purple. So is this correct? No, gram-negative is supposed to be pink. So no. So the problem right now, if you forget to do the decolorization, both your cells are purple. What happens if you forget safranin? So for gram-positives, is that a big deal? No, they're still going to be purple, right? Just a lighter shade of purple, right? So they're going to be purple, iodine, they're going to be purple. We're going to decolorize. They're going to stay purple. Oops, we forgot the safranin. Oh well, they're already purple. They'll still be purple. Is that correct for gram-positives? Yeah, we want purple. Right? And again, they're still going to be purple. You're not going to notice they're not a dark purple. Gram-negatives. They're going to be colorless, right? If you don't stain them again, they're colorless. We're not even going to see them if you forget that step, that last step. So, what if you did it in reverse order? So our gram-positives, what do you think they're going to end up? If we start out with safranin, they're going to be red, then iodine, and I can tell you it's still complexes just like crystal violet. So they're going to stay red in this case when you decolorize them. And you add crystal violet, they're going to become a dark purple. Right? So red and purple, dark purple. Is this correct for gram-positive? Yeah. But for our gram-negatives, if we start out with red, right, add the iodine, we decolorize them, they become colorless, right? Then we stain them in crystal violet, they're going to be purple. So no. We've got purple gram-positives and purple gram-negatives, but I can tell you that the gram-negatives will be a lighter purple than the gram-positives, because I've done it. But you can see the two of them when they're side by side, but if you were to look at them separately, you wouldn't know one's darker or lighter than the other, right? That would be a nightmare. Distinguishing between pink and purple, y'all, by the way, is a nightmare enough. So here's a summary, right? If you make any of these mistakes or reverse the order, you're screwed. Right? You're going to end up with the same color or very slight differences of the same color or not see them at all. If you end up with a pink gram-positive, right, we're ending up with a false negative result. This is not an accurate negative result, right? Gram-positives should not be pink. Or you could end up with false positive results, right, where gram-negatives, which are supposed to be pink, are coming out purple. This is why, again, we'll run controls when we do our unknowns, right? Because you forget any of these steps, you do any of these steps in the wrong order or improperly, you're not going to get the correct results. Your results are going to be inaccurate. And of course, undetermined there, you wouldn't be able to really see them. So both crystal violet and saffron are basic dyes. They can be used for simple staining like we did last week. But in this case, we're using crystal violet and saffronin, and we're going to get a result gram-positive or gram-negative. Why is that possible? What's so special about this technique that we're using today, different from what we did last week? The decolorization step. Removing the color from one group and not the other is what makes this a differential stain, why it tells us different stuff other than just shape and morphology. So the key step is the decolorization step. And of course, this step is enhanced by the iodine. So if you saw large eukaryotic cells in a preparation of your gum line, they were most likely your own epithelial cells. Would they be gram-positive or gram-negative? Well, what makes a bacteria gram-positive? The peptidoglycan. That's its cell wall, right? Do we have cell walls? No. So they're definitely not made of peptidoglycan. So can we trap in the crystal violet iodine? No. We're going to be pink, so we are gram-negative. Doesn't really matter for us, right? Because that doesn't tell us much information, right? We don't stain our cells gram stain because they're all going to come out gram-negative. We stain bacteria with the gram stain because we want to know, are they gram-positive or are they gram-negative? Because it helps us narrow down the possibilities basically in half. 40% of all gram-positive, of all bacteria are gram-positive. 60% are gram-negative. So if you have an unknown bacteria, like you will after midterm, this is going to narrow your possibilities in half. Right? If you have a gram-positive or a gram-negative, the possibilities out there have just been knocked down to about half.
So if it's gram-positive, you automatically know it's going to be eukaryotic cell.
No. If it's a bacteria, it's going to be gram-positive or gram-negative, right? And that has to do with its cell wall. So if it's gram-positive, it has that thick peptidoglycan layer. If it's gram-negative, it probably has that outer membrane. But if you don't know what the bacteria is, right? You know it's a bacteria, you don't know what it is. You don't know if it's Klebsiella or a bacillus, because they're both rod-shaped, right? But bacillus are gram-positive, and Klebsiella is gram-negative, right? So it's going to help you determine what you've got, because bacillus are always gram-positive. Other bacilli are usually gram-negative. Right? So if you don't know what the organism is, if you then know, okay, this one's gram-positive, it narrows the possibilities of all the possible bacteria out there basically in half. See how valuable that is? Right? And for treatment purposes, it's very important, because remember I said gram-negatives can be very toxic for us because of that outer membrane. Right? You want to treat your patient cautiously if they have a serious gram-negative infection, right? If you know that that organism is gram-negative. Make sense? Okay. So this is our scientific method, right, for a gram stain. For the next stain that we're going to do is the capsule stain. So we're trying to identify this extra layer that some bacteria will have. So it's also sometimes referred to as a Glycocalyx, okay? And you could have one that's uniform, which is what we call a capsule, or one that's diffuse, which is called a slime layer, right? But it's always going to be outside the cell wall and cell membrane. So here's your cell membrane, here's your cell wall, or maybe even an outer membrane for gram-negatives, and then you're going to have your slime or capsule layer. Your capsule layer is very defined and is securely attached to the cell wall, where the slime layer is not firmly attached, and it is diffuse. But both of them typically are made out of sugar, so glycocalyx actually means sugar coat, right? They're coated in sugar. There are some few exceptions. There are some that are made of glycoproteins, so sugar and protein. And there's at least one case where it's made of just amino acids, just protein, basically. Why do these organisms have these layers? Why do they have a capsule layer or a slime layer? These glycocalyxes allow them to attach to surfaces, and this can be very beneficial, especially for pathogenic organisms. It allows them to attach to us. And then also, it's an extra layer of protection. And then especially in our bodies, it helps them avoid phagocytosis, destruction by white blood cells. Our cells can't recognize them. It's like they're wearing a disguise, right? They have this extra coating. We can't detect them, right? Not easily. So it's important to know, does the organism have this extra layer? Does it have a capsule layer? And so we have to use a special stain in order to visualize the capsule. The capsule itself is not charged. So most of the dyes that we use are charged, right? They're positive or they're negative. If it's not charged, it's not going to attract those dyes. So instead, we have to stain everything else, right? We have to stain the cell and the background. So we have to employ both of the types of dyes and procedures that we've been doing, right? We've got a negative stain. We've got to use an acidic dye and stain the background. We've got to use a basic dye or a substance that's going to stain the cell so that we can see the unstained clear section around the outside of the cell known as the capsule. So in your book, it tells you to apply serum. We don't do that step. So you'll notice in your guys' handout for today that I have on there that we skip that step. And then we're also going to skip another step that your book tells you to do, and that's to rinse when you get done. Do not rinse your slides. Just tip the excess fluid off and put them in your books and blot them dry. There's a reason for this. It's because we're skipping the serum step, and so therefore when you go to rinse, you'll rinse off most of your stuff, won't stick to the slide. because of the lack of serum, which helps stick it. The serum has a lot of proteins in it, right? We know proteins are sticky. Even though capsules stick to us and other surfaces, they don't stick to glass very well. They're actually kind of slippery. So to keep them from being washed off, we're not going to rinse. Okay, we're just going to blot. So basically we're going to start out like a negative stain and stain the background. And then we're going to do like a simple stain and we're going to stain the cells. But it's also going to change the color of the background. And actually it's a pH change that happens. It's not a staining that happens. So the reagent that we use has acetic acid in it, which is the same thing as vinegar. So if you don't like the smell of vinegar, you won't like the smell of this reagent. So it's going to be a little smelly. But it's not anything that's going to hurt you. It's basically vinegar. So encapsulated cells are slippery. That's typically why procedures will have you use serum to help stick them to the slide. So some oral bacteria produce extracellular capsules. What's the benefit? Why would bacteria in our mouths want a capsule? So they can stick to us, right? That biofilm that develops, what we call plaque, is bacteria stuck to our teeth because of the capsule and the extracellular matrix they'll form to stay stuck. This is why you must scrub. This is why you should floss, because the toothbrush doesn't get in between the teeth. Food does, and bacteria does. You want those guys out of there. You need to physically remove them because they're attached. So, and again, it's protection for them from phagocytosis, detection by our antibodies. It's really good news for them, really bad news for us. So, this is a slightly different stain than what we use, so the colors are different, but same principle. The cells are stained, the background stained, the capsule itself does not stain. It's the unstained halo you'll see around the cell, because remember, it's the outermost covering of the cell if it's present. This is more like what ours will look like, a kind of grayish background and a purplish or pinkish looking cell. Again, these have capsules because you see the clear halo around them. So what is why is the acidic stain used? Remember, what charge do acidic stains have? Negative. So what are we staining when we use the Congo red? The background. This is only the first step. Do not look at a red slide under the microscope. Once you put oil on it, you destroy it and you have to start all over again, right? After that dries, then you've got to add the Menville's reagent, and it's going to turn the slides kind of a grayish color, right? Gray slides are what you'll be looking at under the microscope. So, why are we using the Menville stain, this basic stain? To stain what? We've stained the background. What else do we need to stain? The cell. We need to stain the cell. And again, the pH change is going to change even the background, right? We're going to go from red to a kind of grayish, bluish grayish color. Is the capsule stained? No. And so we've got to stain the cell in the background because we can't stain the capsule. It doesn't get stained. So, that's a capsule stain. Do you see a capsule? No. And I know it's a capsule stain because the cell is stained and the background is stained. But what do we not see? We don't see a clear zone between the cell and the capsule, right? Which means that this one does not have a capsule. One of your organisms today is going to look like this. No capsule. So, if you do just a simple stain, will we see a capsule? No, it's just going to stain the cell. The only reason you know maybe this one has a capsule is because I drew a line around it. But that capsule in the background would blend in with each other and you wouldn't see it. Now, negative staining I found with the Congo red. Did you guys notice that some of them you could kind of see some red on the inside of the cell when we stained the Klebsiella with the red dye? You saw some clear, but you may have saw some red on the inside. Okay. If you just negative stain, usually it's just going to stain the background. But I've found with some of our organisms, especially Klebsiella pneumoniae, which has a really thick capsule that you guys will really be able to see today, that we had some staining happening of the cell itself. How or why, I don't know how that happened. But it's kind of cool. But normally, it would just stain the background, like it did for Micrococcus luteus, right? We just saw clear cells. And you don't know if it has a capsule, right? Because the cell itself didn't stain, so you couldn't see the unstained portion of the capsule. So, our scientific method, right? Why is this important? Because encapsulated bacteria typically are pretty pathogenic for us, right? They can stick to us. Klebsiella pneumoniae, right? I already told you guys, that one's going to have a really thick capsule. It causes a really bad form of pneumonia because it can stick to us. It can evade phagocytosis by our white blood cells, right? It's bad news. But again, nobody's licking petri dishes. You'd have to be exposed to a lot of these guys to get this infection, right? So it's okay to work with it aseptically today. No one's going to get Klebsiella pneumoniae. Okay, infection. But others, you know, like in our mouths and stuff like that, that create the dental problems that we have, again, they're encapsulated. They stick to us. And they avoid destruction when they get into our body by our white blood cells. So here's just some terminology, right, for you guys. I'll post this PowerPoint for you so you have each of the terms written out. And that's it for that. I'm going to do a quick demonstration